A new study from researchers at Henry Ford Hospital found substantial evidence that a regenerative mechanism involving damaged nerve fibers in the spinal cord could have the potential to aid functional recovery by a majority of stroke victims. Due to unsolved mysteries about the brain and how nerves repair themselves, there is currently no known means of restoring damage to this region of the body. To solve some of these mysteries, Henry Ford researchers focused on changes in the axons, the fibers within the spinal cord that control nerve signaling and impact voluntary movement after a stroke. They used genetically modified mice that had their axons stained with a fluorescent dye that was visible under a powerful microscope. The mice were trained for 5 days to use their left paws to retrieve food pellets from a dispenser designed to test their dexterity. They were also given a food fault test to determine how well they could walk on an unevenly spaced grid. Next, the mice were split into 4 groups. In one, the carotid arteries were blocked with a suture for 1 hour. Once the suture was removed and blood flow was restored, they were given additional surgery to cut the axons of the cortical spinal tract. The other groups were either given no surgery or sham surgery. The researchers concluded that in the initial stages following a stroke, improvements in voluntary movement can be attributed to the reduction of swelling in the brain area due to trauma and other spontaneous repairs. However, later improvements were the result of a reorganization or regeneration of nerve cells within the spinal cord in response to changes in the nerve network.